Hello, this is Dr. Mathangi, Associate Professor of Biochemistry. I have 20 years of teaching experience and 15 years of strong research experience in the field of biochemistry. Normally, biochemistry is dreaded as a subject, being volatile, difficult to memorize the concepts. I am here to share my knowledge and help you all learn biochemistry with ease. I am passionate about the subject since my UG days, when I realized that this subject is the foundation for understanding disease pathology and its treatment. And this passion towards the subject helped me ace my examinations both in UG and PG as a topper with a gold medal. I am here to share my knowledge to make the understanding of biochemistry much easier, so that none of you would ever say, I dread biochemistry, I don't like biochemistry, or it is difficult to remember. And let us change the tagline to I love biochemistry. Today we are going to see a session on metabolism of ketone bodies. Metabolism of ketone bodies is also called as starvation metabolism. It teaches us, or it makes us understand how our body and all the cells of our body responds in times of starvation. So in 2025, do we ever starve with all the food delivery apps around? Definitely we do not starve. When we say starvation, we talk about the non-fed state in our body. So when the fuel for our body is glucose, when you consume a diet, 2 to 4 hours following the consumption of the diet, technically it is called the fed state, where the body absorbs the energy and it is supplied to every cell of the body. The remaining energy is stored in the form of glycogen. Whereas when we sleep during the night time, when we go without food for 8 to 10 hours, maybe it is not possible for most of us who open the fridge midnight and eat something, but this is generally considered fasting when we sleep, when we go without the food. It is nearly for 8 to 10 hours. When you sleep, your metabolism does not shut down, and your body, especially the major organs of the body, they need continuous fueling in the form of energy, which is not 100% supplied by glucose. For the glycogen reserves may get depleted. Then what else comes handy for all the major organs of your body to function? So these molecules are called as ketone bodies, and in today's class we are going to learn at length about ketone body formation, which is called as ketogenesis, and its breakdown, which is called as ketolysis. This topic is of utmost importance for UG and PG, and questions do come in PG NEET related to ketone body metabolism. So let us begin the session. So what happens during starvation, or in the state when we are not fed on diet, especially when we are sleeping or when we are going without diet? So that is called as starvation. So there is always a decreased carbohydrate intake, or the supply of carbohydrate is less. So there is depletion of glycogen from the liver and the muscle. So to compensate for glucose, fat breakdown begins in the adipose tissue, and these fatty acids are mobilized to the liver, where they are converted to ketone bodies, and these ketone bodies actually acts as a primary source of energy for the whole body during starvation, which is explained in this flow chart. Well, we all know from childhood that glucose is the first choice of energy for all cells of our body, but other than glucose, when glucose is not available, ketone bodies, amino acids also acts as a source of energy. While the breakdown of protein to give amino acid is the last thing kept in our body, what comes to rescue in the absence of glucose is fatty acid, and these fatty acids are converted to ketone bodies, which are then used as a fuel or source of energy by the major organs and organ systems of our body. So this diagram, it shows the order of preference of fuel source by various body cells. RBCs, we all know they are enucleated cells and they don't have any organelles. So the only choice of fuel for RBC is glucose. For brain, it is glucose, and in the absence of glucose, as in the case of starvation, ketone bodies play a significant role. The third is liver. Liver utilizes glucose as a source of energy. In the absence of glucose, fatty acids are broken down by the liver. The same thing for skeletal muscle, where glucose is used as the principal source of energy. In the absence of glucose, ketone bodies come handy. And also in adipose tissue, glucose is used as a source of energy and fatty acids are stored. So let us see what happens during a starvation. So when you are starving, that is following ingestion of meal, obviously there is a spike in the level of insulin, and glucose gets transported to the liver and the skeletal muscle, and it gets stored as glycogen. Say you fast for nearly 10 to 12 hours a day. When you sleep, at that time the liver glycogen depletes, and there is a newer process that is happening called as gluconeogenesis. Gluco means glucose, Neo means new, genesis is synthesis, where glucose is synthesized from a non-carbohydrate source. This is for a brief period of time, following which ketogenesis would begin. So, ketogenesis is genesis of ketone bodies, and this begins close to 18 hours following starvation. So, what are ketone bodies? The ketone bodies are acetone, acetoacetate and beta hydroxybutyrate. So, the three molecules are shown in the image. Acetone, all the three have a ketone group except for beta hydroxybutyrate, but still it is also considered a ketone body. In a normal well fed state also ketogenesis happen, but the amount of ketone bodies produced under normal well fed state, when a person is not starving, when a person is not diabetic, the normal state of ketone body should be less than 1 milligram per deciliter in plasma. During starvation, it increases exponentially, resulting in a condition called as ketoacidosis. Ketoacidosis is a clinically dangerous presentation, predominantly seen in type 2 diabetes mellitus, and we will see about ketoacidosis in detail in later part of the presentation. Normally, ketoacidosis is characterized when the level of ketone bodies in blood go above 500 milligram per deciliter. Ketone bodies metabolism we study under two categories, ketogenesis and ketolysis. Ketogenesis, the substrate for ketogenesis is always acetyl-CoA, which is the breakdown of fatty acid catabolism. The site for ketogenesis is always mitochondria, and the organ where ketogenesis takes place is liver. The free fatty acids, which are the precursors of ketone bodies, are derived from adipose tissue. Nearly 30% of free fatty acid are drawn to liver for ketogenesis. They first undergo lipolysis, beta oxidation to form acetyl-CoA. These acetyl-CoA cross the mitochondrial membrane into the mitochondria, where they undergo ketogenesis. So, let us follow the chart which shows the steps involved in ketogenesis. The first is the transport of acetyl-CoA into the hepatocytes, and they get into the mitochondria through the carnitine palmitoyl transferase 1 protein. Upon entering the mitochondrial matrix, the acetyl-CoA gets converted into ketone bodies. So, the first step in ketogenesis is combination or combining of two molecules of acetyl-CoA to form acetoacetyl-CoA, and this reaction is catalyzed by the enzyme acetoacetyl-CoA synthase. This molecule of acetyl-CoA gets converted into HMG-CoA. So, this pathway is common even for cholesterol biosynthesis. So, the enzyme catalyzing this is HMG-CoA synthase. HMG stands for hexamethyl-gluteryl-CoA. So, now this HMG-CoA is a precursor for the ketone bodies. So, HMG-CoA is acted upon by the enzyme HMG-CoA lyase. As we all know, lyase is an enzyme which will break down HMG-CoA to form two molecules, one being acetoacetate, the other is another molecule of acetyl-CoA. So, this acetoacetate itself is a ketone body, and this undergoes decarboxylation, which is non-enzymatic, to form acetone. The other acetyl-CoA will get converted into beta hydroxybutyrate. So, this is how your body synthesizes the three types of ketone bodies, namely acetone, acetoacetate and beta hydroxybutyrate. And always remember, the synthesis happens only in liver and inside the mitochondria. Let us go to the next part of ketone metabolism, which is breakdown of the ketone body or the utilization of the ketone body to produce energy. So, that we call it as ketolysis. Always ketolysis happens in the extrahepatic tissue, that is tissues other than the liver, that includes brain, heart, muscle and renal cortex. Liver and RBC never undergo ketolysis. This should be kept in mind. RBC does not have any organelle, does not have mitochondria, so there is no ketolysis in RBC, and liver is the producer of ketone bodies, so it never utilizes ketone bodies. So, ketolysis, it is transported to different parts of the body from the liver is what we understand now, and the transport is easy because ketone bodies are water soluble. So, they are transported in a dissolved state through plasma to different parts of the body. It could even cross the blood-brain barrier as they are small in size and also water soluble. So, ketone bodies provide 75% of energy to the said organs during starvation. So, this image, it shows the transport of ketone bodies from the liver to various organ system through blood. Going to ketolysis, where the ketone bodies are utilized by the extrahepatic tissue to produce energy in the form of ATP. So let us go through the metabolic pathway. The first step in breakdown of ketone body is, if we start with beta hydroxybutyrate, it gets converted into acetoacetate by reduction. The enzyme which catalyzes this is beta hydroxybutyrate dehydrogenase, and for this process NAD is required and one molecule of NADH is released for every molecule of beta hydroxybutyrate that is converted to acetoacetate. So there is yield of energy in the first step. Secondly, this acetoacetate gets converted into acetoacetyl-CoA by activation by the addition of CoA unit. Followed by this, it will be acted upon by an enzyme called as thiolase, where the end product is acetyl-CoA that will go into TCA cycle. So if TCA cycle is blocked, technically till the stage of acetoacetyl-CoA, there will be production of 3 ATP, and 1 ATP goes for the activation of acetyl-CoA. So if we look into the energetics carefully, 2 ATPs are produced till the stage of acetyl-CoA, and if TCA cycle is complete, 24 ATPs will be produced. Adding 2 to 24, 26 ATPs will be yielded by the breakdown of one molecule of beta hydroxybutyrate. As we all know the energetics of glycolysis and TCA cycle, this yield is much lesser than what is being given by glucose, but still this helps the body to meet the energy requirements in the absence of glucose, especially during starvation. So going to the clinical significance associated with ketone body metabolism, let us get introduced to the three terms. First is ketonemia. Rise in ketone bodies in blood above the normal level is called as ketonemia. Please recall the normal level of ketone body in blood is 1 milligram per deciliter. So anything over 1 milligram per deciliter is considered as ketonemia. Next term is ketonuria. So when the level of ketone bodies increases in blood, obviously it gets filtered through the kidneys and they get excreted through the urine, and the condition of excretion of ketone bodies in urine is called as ketonuria. So the excretion of ketonuria itself is abnormal in urine, please make a note. The third condition, the clinical significance, is called as ketosis, where there is accumulation of abnormal amount of ketone bodies in the tissues and body and also in body fluids, called as ketosis. And the fourth condition is ketoacidosis, where the pH of the plasma becomes acidic because of the accumulation of excess ketone bodies. So before we close this session, let us recall the learning outcomes. So after listening to this lecture, you must be comfortable recalling the names of ketone bodies, describe the site where they are synthesized, and the mechanism or the steps involved in both ketogenesis and ketolysis. And you should also be comfortable describing the energetics, describing the yield of ATP per molecule of beta hydroxybutyrate that is broken down completely, and also be familiar with the clinical implications associated with ketone bodies, namely ketonemia, ketonuria, ketosis and ketoacidosis. So in conclusion, we could say ketone bodies are the guardian angels, for they are the alternative source of fuel to your body in the absence of glucose. I hope this session made your understanding of ketone metabolism better. If you have any queries, please write to us and we'll be more than happy to answer your queries. Thank you. Happy learning biochemistry.